Hi everyone, my name is Josh and welcome to my poster/flash talk on the applied use of single-cell RNA sequencing in studying immune microenvironments. I'm a current first-year PhD student at the University of Wisconsin-Madison and the DIN Lab. So a brief introduction of what we're interested in is looking at tumor immunology more holistically rather than just the malignant cells. The interaction between stromal tissue, immune cells, and malignant cells correlate with both survival outcomes and available treatment options. In this diagram, two types of immune landscapes are represented. On the left is a desert phenotype with no immune cells present, and on the right we have infiltration of immune cells, including cytotoxic T cells that are responsible for tumor clearance. So the particular cancer we're interested in is high-grade serous ovarian cancer. It's been kind of mysterious and sadly a really devastating disease. Recent evidence has suggested it actually originates in the fallopian tube. So leveraging this information and modern technologies such as single-cell RNA-seq, we can look at the immune and stromal compartment involved in the healthy fallopian tube and understand what changes during cancer progression. So the data set I'm using represents around 53,000 cells from 8 different donors. And my work is to focus on the box cells here, representing the immune and stromal compartments of the fallopian tube. And the plot shows a dimensional reduction using UMAP used to broadly annotate the cell types. So from the broad dimensional reduction, I take out the cells belonging to a specific compartment, and here I subsetted out the fibroblasts and other stromal cells to reanalyze with clustering, and doing so I've identified 7 different subsets. Differential expression analysis was used to annotate these cell populations, and what we find are these 3 classical fibroblast populations as well as some others. Fibroblasts regulate the extracellular matrix, and functionally these cells are just as activatable as immune cells. They can secrete different cytokines that regulate the inflow and outflow of other cells. So what was interesting is finding a population of complement expressing fibroblasts, and complement is just a protein as part of the innate immune system that works to kill pathogens through pro-inflammatory enzymatic cascade. And the main goal of this work is to bridge the healthy cell types from the fallopian tube, and for which of these cells contribute to actual malignant samples. So one method is comparing single-cell data to these large public data sets, such as the Cancer Genome Atlas. And here, bulk RNA samples are differentiated by these molecular subtypes. These represent the tumor and non-tumor cells within a given sample. And obviously, bulk data on its own cannot show the true composition of sample types since it's pooled data. However, in the future, I'll be using a computational method known as deconvolution to estimate the proportion of the cells using my single-cell data as a reference. So enrichment score is kind of an easy workaround before diving into deconvolution, and it looks at what cell types may be overrepresented. On the left are the same expression results from the differential expression analysis from stromal clusters. I take the top ranking genes and use that to subset the same genes from the bulk RNA data. On the right are the results of these enrichment scores, separated by cluster. And what we see is that in the mesenchymal subtype, it's highly enriched with the complement expressing fibroblast population. In our other cell subsets, such as the T and NK cell group, we see the majority of these cells, all this here in red, pink, and orange, are cytotoxic T cells, representing the dominant cell type. And since this tissue is non-malignant, I believe the lack of immune activation is the reason why we can't separate beyond our broad annotations of CD4 and CD8. But we did identify 2 natural killer T cells, or natural killer subsets, which are a part of the innate immune system, and they're differentiated by their use of granzymes, which is the enzyme they release to actually kill malignant or infected cells. Immunology itself is just hugely complicated because of the number of interacting cell types that define the microenvironment. So modern therapies exploit these interactions to modulate activation states, in a way that favors tumor clearance through immune cells. And I detect these known ligand receptor pairs by subsetting them out of our count matrix and using the metadata from the clustering results to do this by cell-by-cell basis. And this is a clustering for the entire data set. On the vertical axis, we have our sending signals, and on the horizontal, we have our receiving cell types. The number of the box represents the unique number of interactions possible. And overall, we see that the stromal cells, and particularly the fibroblasts, have a huge number of potential interactions with other cells in the microenvironment. And what I hope to do with this in the future is detect the global change of communication after integrating this single cell data with malignant tissue. So in brief, the work I talked about characterizes non-epithelial cells in a variety of ways. And the ones that have shown are differential gene expression analysis and cell-to-cell communication. This work has suggested some fibroblasts may have a potential role in the mesenchymal subtype of high-grade serous ovarian cancer, and warrants further investigation of this subset as a biomarker to evaluate prognosis, since the mesenchymal subtype is known to have the worst survival outcome. In the future, my work will evaluate different deconvolutional methods using my single-cell reference to infer cell proportions of bulk RNA samples. I'm also working with collaborators that have collected true malignant single-cell samples, and this will be integrated within the dataset to determine cluster-wise how these cells are differing. And with that, I'd like to thank those that have helped me, and I appreciate you listening in.